Perfect. Hello, everyone. Good afternoon or good morning, depending on where you're joining us from. My name is Alicia Gallier. I am Business Development Manager at One Nucleus, and I'm delighted to open our DomainX webinar today. So One Nucleus is hosting the DomainX webinar, and the topic we're discussing is Director Biology is a powerful approach to accelerate hit finding, concept and application to drug discovery programs. So before we go into the content side of things, it's my job just to go over some brief housekeeping rules. I know we're all very used to the virtual world now, so I'm sure you're very familiar. But just kindly, if you are not speaking, if you can remain muted and cameras off, that would be appreciated. As you will have just heard, we are recording the session. So the aim is that we can make the recording available post-webinar, and there'll be a communication that goes out afterwards with more details on that. Also to let you know, if you haven't located it already, there is a chat box enabled. So we do want this to be an interactive session. So throughout the presentation, please do feel free to drop your questions and comments into the chat box. And then afterwards, it's my job to deliver those to the speakers online, and we'll try our best to get to them and answer as many as we can. So yeah, don't be shy and use this as an opportunity to communicate and link up directly with the experts that could help you in your own drug discovery challenges. So before we go into the main presentation, I'm just going to play a short video. So Natalie Winfield, who is Executive Vice President, Commercial Operations at DomainX, has done a short intro to DomainX for us. So if you just bear with me, I'm going to share this video and play this now. Thank you. Hopefully everyone can see that, and I'll start to play that.
Hello, thank you all for joining this webinar. I'm Natalie Winfield, Executive Vice President of Commercial Operations at DomainX. I'm going to give a very quick introduction to DomainX before we get into the science. So for those of you who don't know much about DomainX, we're a fully integrated provider of preclinical drug discovery services. We're based in the Cambridge, UK area and currently have 2 sites. Our site at Chesford Research Park houses our chemistry and bioanalytical departments, and we opened a new site at Unity Campus for our biology department at the beginning of 2024. We have received multiple awards for our services, including most recently the prestigious King's Award for Enterprise. We have a world-class team and currently have over 100 staff. Our scientists are highly qualified and experienced, with around 90% having either a master's degree or PhD. They have a wide range of expertise across disease areas and target classes. We have a global client base and work with a range of different types of companies, including biotech and pharmaceutical companies, academic institutions, and patient foundations. We offer the full range of preclinical drug discovery services, including protein production, assay biology, medicinal, computational, and analytical chemistry. We support our clients through all the stages of drug discovery, from target validation and protein production through to lead optimization. If you would like to find out more, please do get in touch. I hope you enjoy the webinar. Thanks.
Excellent. So I hope everyone could hear that okay. Just to let you know, Natalie is online and she will be able to help pick up any of the questions at the end of the session as well. So with that, I don't think you need to hear too much more from myself. So I'm now going to hand over to our speakers. So we have Joe Mason, Associate Principal Scientist, and Nick Bland, Group Leader at DomainX. And so Joe is going to be sharing the slides. So Joe, I'm just going to make you spotlight you here. Okay. And so over to you, Joe. Thank you.
Thank you very much. Hopefully you're able to see the slide deck in the correct presentation mode. Excellent.
Looks good. Thank you.
Thank you very much. So welcome everyone Today we're going to be talking about directed biology and what it can do for drug discovery programs. So by quick way of introduction, my name is Joe Mason, and I'm an associate team leader in the chemistry department at DomainX. I'm our high throughput chemistry specialist, and I'm responsible for building and maintaining our capabilities in high throughput chemistry and directed biology. Prior to working at DomainX, I spent 5 years at GSK in the medicinal chemistry department, again with a focus on synthesis and high throughput chemistry. D2b and directed biology is very much a collaborative exercise between assay and biology and chemistry. And joining me today, I've got Nick.
Yeah, sorry. So my name's Nick. I'm a group leader in assay biology at DomainX. I've been here since 2020. I've got almost, just over 20 years experience in fundamental research and drug discovery. And the projects I've run have covered the gamut of assay biology from cell biology to biophysics, although I have a particular expertise in entomology. Since joining DomainX, over the last couple of years, I've been working with Joe to get our D2b pipeline set up, and so looking forward to talking to you about it.
Thank you, Nick. So as I say, today we're going to be talking about what directed biology is and how it can work in a drug discovery program. I'll start off by covering what it is, some of the literature background that underpins modern D2b experimentation. I'll go through a typical D2b workflow, and then we'll cover some frequently asked questions. I'll then hand over to Nick for his perspective from assay biology and to discuss a recent in-house case study on the development of PROTACs. So in answering the question, what is directed biology? Let's think about for a moment this hypothetical drug discovery project that begins with the lead compound shown here. When we do this hypothetical drug discovery project, we might want to explore the structural activity relationship, or SAR, of the amide vector shown on the right-hand side, using a range of commercially available acids. And to build that SAR, we'll want to explore a large chemical and property space. We'll want to use all available tools to ensure that we make the right compounds. And of course, we want to be efficient with both our time and our resources. Now, given the vast number of commercially available carboxylic acids, it's reasonable to expect, or even likely, that by the time we've enumerated and filtered using rigorous criteria, we still end up with about 1,000 compounds. Each of these compounds would be considered drug-like, and in an ideal world, we would want to synthesize and test each and every one of them. With traditional chemistry, though, we quickly come into this purification bottleneck. This alone would prevent us from synthesizing and testing all of the desired compounds. And we'd have to go back to our selection, and find more arbitrary methods for that selection. But what if we remove that purification bottleneck? When we remove the purification bottleneck, we're now able to synthesize the full set of compounds in a matter of days rather than months. We carefully design D2b compatibility experiments to ensure that the assay data we get is meaningful. We cherry pick the top hits for compound resynthesis, purification, and testing to ensure that the assay data we collect is validated. By using D2b, we avoid wasting time and resources on compounds that look good on paper, but prove to actually be inactive. We still resynthesize, purify, and retest the very best compounds so that we can be certain that our next scientific hypothesis is built on a solid foundation of data. By using D2b in the right way, we can accelerate our drug discovery programs without compromising on the quality. At DomainX, we consider D2b to be akin to a filter, where we can reduce large design sets from thousands to a more manageable size for purification and synthesis. We still invest that time to design the compounds and use computational tools to aid us where possible. It's not about throwing the kitchen sink at it. It's a pragmatic answer to the question of the vast chemical space that's always available to us. Our D2b readouts include potency, chrome D, EPSA, and we're currently validating a microsomal stability assay for crude D2b compounds. As a result, our D2b-enabled drug discovery programs invest those purification efforts only in the right compounds with the right measured properties. And it's not just us. we take a look back at the literature, we can find reports of D2b going back all the way as far as 2003, where off-rate screening was used as a measure of potency, and there's some notable contributions from the Cernak lab. Fast forwarding to the past 4 years, we find contributions from the Domlin group, and application of D2b to covalent fragments by GSK. More recently, we find application of D2b to PROTAC synthesis with its inherent synthetic challenges, and that's followed up with a bolus of papers in 2023. Notable contributions here come from AstraZeneca, GSK, the COVID Moonshot Consortium, and again, the Cernak labs. Molecular glues make an appearance in this space as well. These compounds, which are notorious for finding that balance between inhibitor versus degrader, is a great one for D2b, as it brings that acceleration in terms of the rate of SAR generation and makes it really well suited for molecular glues. And then most recently, in 2024, we see yet more examples in the PROTAC space, and a really nice example from Merck, where they found excellent correlation between crude and purified libraries of HDAC inhibitors in their microsomal stability assay. For projects where D2b can be applied, the advantages are really quite striking. We can reduce the time taken to synthesize those compounds from a matter of months to days, and we reduce the amount of starter material required for those syntheses quite dramatically as well. This in itself comes with some cost implications, some positive cost implications, and that's not to mention, of course, the solvent and the environmental benefit that comes from removing all that solvent associated with the purifications. I'll now change tack slightly and discuss our D2b workflow in a step-by-step process. So we begin, like most projects, by developing either a biophysical or biochemical assay that we're going to use to test our D2b compounds. A number of different assay technologies have already been validated for D2b, and Nick's going to cover a few of them later on in the talk. Once that assay has been developed, we'll then look to conduct a D2b compatibility test to ensure that the crude reaction samples are tolerated in that assay. We do that by taking project-agnostic crude reaction samples into the assay and to show that they have no effect in that assay, i.e. they look like the DMSO control. By project-agnostic, what we mean by that is that both the starter materials and the products of that chemical transformation are structurally separate to the lead series for that project. We then take those same crude reaction samples and spike in a positive control to show that we can replicate the published activity of the purified control. In parallel with this, we move on to step 3, where we look at reaction optimization. This optimization is carried out in a plate-based format and uses UPRC-MS to determine what's a successful reaction. We then move on to step 4, D2b synthesis. And this takes a Venn diagram intersection between the reactions that work to give the desired product and the reaction conditions that which were compatible in the assay. Again, those reactions are monitored by UPRC-MS, and successful reactions are taken into the assay. This then takes us to step 5, hit resynthesis. Once we've identified the hits from the D2b, we'll then look to rank them using, where possible, a multi-parameter optimization function that includes potency, chromolog-D, EPSA, and metabolic stability. We'll then resynthesize and purify those hits, and then retest them in the assay to ensure that they were true positive. Worth noting that D2b can be an iterative process, so the results from one D2b round can feed into the design rationale for the next D2b round. Alternatively, you could have multiple vectors on a lead compound that you want to explore with D2b in parallel. As such, it's worth noting that D2b isn't necessarily only for very early phase drug discovery projects. If the hypothesis is right, you can use it both at the early phase, hit finding, all the way through to lead op. D2b has already been applied to a number of different drug discovery modalities, including small molecule inhibitors, fragment-based drug discovery, bifunctional protein degraders, and molecular glues. It's not just limited to amide couplings either. You can use reductive aminations, palladium catalyzed cross couplings, and photochemistry, all of which have either been demonstrated in-house or externally in the literature. I've got one example here aware of a successful D2b compatibility test. This example uses spectral shift, a biophysical assay which Nick is going to discuss in a little bit more detail in a later slide. But simply, this assay technology was applied to this adenosine A2A receptor, and we found that the crude reaction samples themselves had activity very similar to this DMSO baseline shown here. When we take combinations of the crude reaction mixture and a known positive control, we find similar activity to the purified control itself. And this is exactly what we're looking for when we talk about a successful D2b assay compatibility test. I think it's worth noting here that, contrary to many commonly held assumptions in the literature and in the field, palladium reactions are tolerated in these assays. You see here SR, which stands for Suzuki reaction, or BR for Buckroyd reaction, again show excellent validation in this single shot compatibility test. There are cases, though, where D2b might not be suitable. It's not a perfect tool, and it's not to be applied to every occasion. For example, you might have a particularly sensitive assay which isn't compatible with the desired reaction conditions, or the medchem hypothesis might not have a well-defined vector to enumerate new analogs, or that hypothesis might require fewer data points than D2b can provide. In any of these cases, it might be more suitable to pursue a traditional library synthesis or another means instead. To efficiently carry out our workflow, we need the right combination of hardware, software, people, and processes. At DomainX, we use a combination of Dotmatics, Microsoft Office, and software generated in-house to design the experiments, facilitate that data acquisition, and then capture those digital outputs. In the lab, we combine these pre-dispensed commercial libraries in a 96-well format with automated liquid handling. This can be examples such as the Thermo Fisher multi-drop or the Beckman Biomech, but we also use the handheld multi-channel pipettes as well to do this. For reaction plates that need heating, we use a standard heater stirrer or heater shaker plate and use a custom metal template with a silicon pad to ensure efficient heat transfer to the wells. For the reaction analysis, we use a Python script that I've developed specifically for the analysis of high-throughput chemistry and D2b plates. This is known as PyPass, and what it does is it allows the chemist to remain in control and scrutinize the results and conclusions that come out, but it automates that tedious and more time-consuming step of assigning UPRC MS peaks to the desired products. The other advantages that come with using the Python script like this is that we get heat maps, annotated chromatograms, and CSV exports as a matter of course. One field where D2b can be particularly advantageous is the field of PROTACs. So PROTACs are biofunctional molecules, for those who haven't seen these before, where one end binds the protein of interest and the other end binds an E3 ligase. The two ends are connected by a linker, and this makes the PROTAC as a whole. In the biological system, once the E3 ligase has been recruited and is in proximity with the protein of interest, that protein of interest then gets ubiquitinated and then directed to the proteasome for degradation. Traditional PROTAC synthesis is quite time-consuming, quite laborious, primarily in part to the size and the complex nature of those PROTACs. In a D2b setting, though, we can take a toolbox of partial PROTACs that combine a variety of E3 binders with a variety of different linkers, each of which have a reactive functional group at the end, ready to connect to a protein of interest binder. As a result, at the outset of a PROTAC project, we can take that pre-plated and diverse library of partial PROTACs, combine it with one or more protein of interest binders, and end up with a library of different PROTACs ready for hit finding. Our partial PROTAC library combines different linker lengths, different rigidities, a number of different E3 binders, and we've got a focus for the cereblon recruiters for those with enhanced near-substrate selectivity. So far, in this first half of the webinar, we've discussed how directed biology can rapidly accelerate that structure-activity relationship without compromising on quality or scientific integrity. We've discussed how D2b can be akin to a large filter, for large design sets, reducing down to a manageable size. And we've shown, hopefully, that it can be advantageous for all drug discovery programs, whether you're looking at small molecule inhibitors or more large and complex PROTACs. So before I hand over to Nick, I'll just cover a few of our frequently asked questions that we commonly get. So to start, can D2b only be used for amide couplings? Hopefully you'll agree, we've seen already, that no, that's absolutely not the case. It can be used with palladium catalyzed transformations. These are compatible with the assays as long as you use the right type of palladium conditions. One question we get is, can it only be used for degraders like PROTACs and molecular glues? Because only these compounds can have a catalytic mode of action, and they're therefore perhaps less sensitive to variability in the assay concentration. And the answer is no. Again, we can use D2b for the finding of new small molecule inhibitors. We always aim to try and get to perfect reaction conversion, of course, and that's why we do our reaction optimization phase in the project. But we will sometimes get incomplete reaction conversion, and this will lead to a lower than assumed assay concentration. What we find, though, is that it's usually within the assay error. For example, 50% conversion is still just 0.3 less than log scale. So we can still obtain that rank order ready for hit resynthesis. One question is, will I have to make or test the same compound twice? And the answer is yes. Yes, for those compounds that get resynthesized, that will be a second time it gets made. But the advantage here is that as an integrated chemistry and biology product team, the overall time to find those multiple hits is substantially lower. One question we get is, how do we optimize the chemistry? And is there risk of finding conditions that are too specific for a single compound? So what we do is as I mentioned, we use plate-based chemistry to optimize the reaction. And we'll not just look for a single product, but we'll look at multiple products when we're doing that reaction optimization. What we can also do is to give ourselves an extra chance of success, is use 2 or more reaction conditions for each product, and then we can pick the best well that goes into the assay on a case-by-case basis. Can you filter the D2b samples? You can, and there is literature to describe filtering D2b samples before introducing them to the assay. But to achieve our rapid turnaround times, and we use approximately 0.075 of a milligram starting material per reaction, we avoid filtration steps where we can to avoid introducing errors. There are cases, though, where an assay might require some filtration, might be incompatible with reaction conditions, and there we might typically switch to more traditional library synthesis. And then finally, how do we avoid introducing oxygen to air-sensitive reactions on these 384 well plates on such a small scale? And the answer is that we use our state-of-the-art glove box, and that reduces the oxygen and water content to less than 0.5 ppm. I'll now hand over to Nick for a discussion on the PROTAC synthesis. Thank you very much.
Thanks, Joe. So I'm going to take you through a case study of a D2b project that we use to identify PROTAC for aurora kinase A. So Joe, if you want to move on. So aurora kinase A is a serine threonine kinase that's got an essential role in controlling the cell cycle. It's involved in multiple processes such as centrosome separation and mitotic spindle formation. And if you inhibit aurora A, you get arrest at the G2 stage. So the G2 stage is after the genome's duplicated, but before the cells have gone through mitosis. So in the G2 stage, the cells are very DNA rich. And you can quantify that using a flow cytometry assay. Flow cyt assays for cell cycle analysis are something we offer as an off-the-shelf solution at DomainX, and we can see some of the data in these panels on the right-hand side. So these histograms summarize that flow cyt data. On the x-axis is the signal from propidium iodide, and propidium iodide is a DNA dye, and the y-axis is just the number of events or cells. In the vehicle controls, you can see that the majority of the cells have got a very low signal, showing that they're in the G1 stage because they've only got one copy of the genome. And then some of those cells, though, are in the G2 stage because they're just about to go through mitosis. If you add the aurora A inhibitor elicitab, you see a shift to having far more cells with a very high signal of propidium iodide because they're all trapped at that G2 stage. Just want to go to the next slide. So you can modulate aurora A using small molecule inhibitors, but there is also precedence for PROTACs. So there is a literature compound called JB170, which we can see the structure here in the bottom right-hand corner. This PROTAC has got a head group that is elicited, the Aurora A inhibitor, a flexible linker, and then the molecule thalidomide. Thalidomide is a binder of the E3 ligase cereblon. And so for this project, we took a small subset of our PROTAC toolbox that focuses on cereblon binders and ran through our D2b workflow, which is on our next slide. So Joe went through this earlier on, and we're going to skip through steps 1, 2, and 3 and just focus on 4 and 5. So in these steps, we've made our D2b plates and we're running them through our primary assay to identify some hits. And then we're going to resynthesize, repurify, and further characterize those compounds. So how do you drive a D2b campaign? Well, it's a very flexible approach, and that's compatible with all sorts of assays from cell biology, biochem, and biophys. At DomainX, we've got a strong biophys suite, and one of our favorite technologies is spectral shift, which is Nanotemper's latest generation of binding assays. So the way that spectral shift works is that it relies on the phenomenon that the microchemical environment of a fluorophore affects its emission spectrum. If you label your protein with a fluorescent dye and then introduce a ligand, the binding of that ligand can affect the microchemical environment of the fluorophore, causing a shift in the spectrum. If you measure the fluorescence intensity at 2 wavelengths, in the case of the dianthus, that's 650 and 670 nanometers, you can find the ratio of those two signals to get a measure of the amount of compound that's bound to your protein. If you plot that out as a concentration response curve, you can calculate a KD, which you can see in this bottom right-hand panel, and that shows some binding of a peptide to a viral protein. So we go to the next slide. So one of the things that we really like about spectral shift is that it's a very quick assay to both develop and run. With a well-behaved protein, you can turn around the assay development in a couple of days. And as it's a 384 well plate-based method, you can run it at roughly medium throughput. And that allows you to screen hundreds to thousands of compounds a day. So we ran a single concentration screening of our D2b plates, and what we found was that almost all the compounds bound, which was not a great surprise, as all these PROTACs have elicited as the head group, and elicited is a very potent inhibitor of Aurora A. So we needed a second assay to triage our compounds. We just go on to the next one. And we chose a cellular degradation assay, which is a very popular approach in PROTAC projects. There are lots of ways of measuring the cellular degradation of a protein, but a very popular format is the HiBit assay from Promega. So HiBit is an 11 amino acid tag that on its own doesn't really do anything, but in the presence of another protein called largebit, they can form a functional luciferase, and you can measure the luminescence from that luciferase as a way of quantifying your protein in the cell. Now, if you tag your protein with HiBit and then you introduce a PROTAC, that protein should be ubiquitinated and should send for degradation, and so that luciferase should never fully form, and as such, you won't get any luminescence. And so we can use that as a way of measuring protein degradation. So we put our compounds through an Aurora A HiBit screen, and what we found were 3 compounds that sort of leapt out as being of interest. They had properties that were comparable or better than JB170 in this assay, and so we consider those compounds of interest. They're all binders by spectral shift, and so we progress those for resynthesis and repurification. One of the key steps in a PROTAC project is demonstrating ternary complex formation. So what do I mean by ternary complex? So in most drug discovery, we look at binary complexes, and that's very simply one ligand, one receptor. You can imagine, say, captopril binding to angiotensin converting enzyme. But for a PROTAC to be a degradation-mediated compound, you have to have a complex of 3 different components: the protein of interest, your PROTAC, and the E3 ligase. And so confirming that those 3 things can come together is essential to proving that you really have made a PROTAC. Next slide. And so how do we do that? Well, we went back to our spectral shift assay, and what you can see here is both binary binding and ternary complexes. So we've got concentration response curves of our PROTACs against Aurora A, a fixed concentration of Aurora A on its own. And you can see that in those curves on the lower half of the graph. But when you add in a fixed concentration of cereblon, that spectral shift flips to the other direction. So from red to blue, or blue to red, depending on which way you're looking at it. And that demonstrates that we can form a ternary complex. We can recapitulate that complex in vitro, which is essential for a PROTAC to be functional. So we've shown that we can form a binary complex, a ternary complex, and we can show that the protein is degraded in the cell, but does it actually have a biological function? Next slide. And yes, they do. So the data on this slide is using the flow cyte assay that I described on my first slide. And what you can see is that when we look at all 4 PROTACs, including JB170, that in a concentration dependent manner, you get an increase in cells at the G2 stage and a decrease in those at the G1. The S phase is actually really quite hard to quantify, so that's why it's a bit of a flat line. But what we can say is that using DomainX's PROTAC toolbox and taking a D2b approach, we've successfully identified some novel Aurora degraders. So just move to the summary. So yeah, in summary, directed biology is a flexible and efficient approach for rapid hit finding, as it reduces your design sets and removes that purification bottleneck. It does require careful validation that your chemistry is compatible with your assays, but if you do that, you get very highly informative data with unparalleled speed. It is a very flexible approach, and you can use lots of different experimental outputs from biophys to cell assays, and it's applicable to pretty much all modalities, fragments, small molecules, but it is particularly well suited to PROTACs, and that's why we combined it with our toolbox and our D2b platform to identify these novel Aurora degraders. And so that just leaves me to say thank you from Joe, thank you from me and all the team at DomainX. So thank you for your attention.
Thank you.
And now we'll take any questions.
Okay, thank you very much indeed, Nick and Joe. So I'm just bringing myself back to the screen. 1 second. Joe, did you want to stop sharing your screen a second just so I can, sorry. Yeah.
Of course.
Perfect. Thank you so much. And then I'll bring myself. So as I said at the start, I have enabled the chat box. So now is your chance to drop any questions in there and speak directly to our experts. Apologies, I'm just having issues on. Here we go. Okay. Okay, I've lost Joe now. Okay. So I'm just looking, let's see if we got any so far. Okay, we've got a couple coming through. So I'm going to read them out to both yourself, Joe and Nick, and then you can decide between you who wants to tackle them. So Helen has asked, what is the false positive rate in your experience, i.e. apparent hits from the D2b process that do not repeat when synthesized in the traditional manner?
Nick, do you want to take that one?
Yeah, go for it.
Yeah, it's a really good question. I've typically, in my personal experience, found it to be probably less than 10%, less than 5%. Normally, what we find is that because in the D2b assay, we've taken in compounds that didn't have a perfect yield, a perfect conversion to the desired product, and so that assay concentration is ever so slightly less than what we've assumed it to be. So when we resynthesize and purify the compound, we then actually test it effectively the correct concentration. And so we find actually the activity generally across the board actually improves when you go to that hit resynthesis. But there are compounds that will occasionally fall out. And usually what we'll try and do is kind of pick out, see if there's any common trends in terms of which ones have proven to be false positives and see if we can identify why that might have happened.
Yeah, and one of the reasons why we run these compatibility assays is to really try and minimize that before we do any of the experiments at all, because false positive rates are going to be very assay specific. You know, some proteins are going to be picking up, you know, heavy metals and things like that much more readily than others, and so that's why for every single assay you've got to really do the compatibility every single time. So far we're having really good results in terms of cytotox, high bit, so for luciferase-based assays, spectral shift, and also we're now doing GCI, which is a chip-based biophysics binding assay, which is also one of DomainX's core biophys techniques. So far, so good.
Thanks both.
Oh, great. Thanks, Helen. Thank you. Okay, so we have a question from P. McIntyre. Can you expand on the flipping of the spectral shift ratios between binary and ternary complex?
Well, so the flipping of the whether it's red shifted or blue shifted doesn't actually really mean anything. It just is an artifact of how the ligand binding has affected that local environment of the fluorophore. What's really nice in this example is that the change when you form the ternary complex is so dramatic that it shifts it from blue to red or red to blue, depending on whether you're talking about your ternary or binary complex. So it doesn't really mean anything per se, but it does mean it looks quite nice on a graph.
Excellent. Thank you very much, Nick. And then looking down, we have one more come through from Gajan. Apologies if I'm not saying names incorrectly, and also apologies if you don't want to be identified. Please just message me directly. So does residual PD in the cross-coupling reactions not interfere with the biological assessment?
Nick, if I take that one, perhaps.
Sure, go for it.
I'll come back to Nick's point around the D2b compatibility assay. So we run that up front to ensure that that palladium content doesn't interfere. As I noted in that spectral shift example with the adenosine A2A receptor, there are cases where you can get a great result. There will be cases, I'm sure, with more sensitive and more picky assays where it doesn't tolerate that palladium. What we're doing at the moment in-house as an internal project is to assess our common reactions. So a Suzuki reaction, Buckwell reaction, amide coupling, these kind of things. It's kind of common go-to medicinal chemistry transformations and assess them again in our kind of go-to assays, if you like. So your hybrid assay, GCI, spectral shift, etc. To kind of learn up front before we go in for a real project, if you like, to determine what's going to be suitable. Broadly, we found them to be quite suitable. As Nick says, so far so good. We've not had any real issues, but that's why we run the compatibility test, and not just once for that chemistry with that assay, but also we'll run it when that new project comes in. We'll run a new compatibility test specifically for that project, so we take into account any differences in cell type, assay type, controls, etc.
Absolutely, and as Joe was saying, everything is going to be assay specific. There's going to be instances where a particular detection technology is not going to be compatible with the palladium, but lots where it's been no problem at all. So we haven't had any problem with it interfering with the dyes in spectral shift, for example, which was something that when we were going into this, I was quite worried about, but actually I was proven to be worrying about nothing. And similarly, as for all these cell-free assays where we're using recombinant proteins, different proteins are going to have different response to the different reaction components. So that's why we do it every single time, so that we go into it knowing whether it's going to be an issue and we can exclude those, or we just find a new assay, and that's what we do. If at first you don't succeed, we'll come up with just a different, better solution.
Excellent. Okay, so I have another question sent directly to me. So can D2b be applied directly to cellular assays?
Yeah, absolutely. So some of the data I showed today was hybrid assay. So that's a whole cell assay. As a counter screen, we run a cytotox just to check that the reaction components aren't killing the cells, and so that we get false positives from a reduction in fluorescence for that. Luminescence for that reason. But there's absolutely no reason why it can't be expanded out to other reporter assays or anything really beyond not just cellular degradation assays. But yeah, we would test it every single time. One of the things that we found is that cell type can have a bit of an effect on whether compounds are cytotoxic or not. But actually, that was the fully purified protests were more toxic against THP1s than against MALT4s. So, as Joe said, we have to characterize it every single time so we don't go down the kind of blind alley that Helen alluded to earlier.
Thank you, Nick. So thanks all for your questions so far. So before we came online, Nick and Joe, we were talking more about your ProTac toolbox. So, you know, you highlighted how you're helping to accelerate research in that area. Is there anything else you wanted to add to that? Any further details you can share with the audience?
I guess primarily it's, you know, I'll highlight it's a really quite diverse collections. We've got multiple E3 binder types. We've got both VHL and cereblons in there. It's a pretty diverse set of linkers. So when the team are putting it together, they scour the literature, pulled in all the kind of common linkers, the ones you might expect, but also some of the less common ones, some of the newer linkers that have been recently published. And then again, I'll kind of highlight likewise with the E3 binders, particularly with the cereblon. We've looked to kind of come away from your traditional kind of thalidomide that is in there, of course, but we've looked at other bits and pieces as well. So it's a newer cereblon binders to kind of augment that library.
Thanks, Joe. Excellent. And in the meantime, I've got a couple more questions come through. So David said, do you ever see issues with unreacted components of the reaction interfering with the readout? You might imagine that for PROTACs on reacted, Pol ligand, E3 ligand could outcompete the PROTAC. Sorry if I didn't read that correctly across.
I get it. And you're
Yeah.
absolutely right. So for a simple binding assay like spectral shift, that is more of an issue. Whereas for a PROTAC, when you're actually seeing degradation, assuming it truly is driven through a E3 dependent mechanism, you would have to have a lot of unreacted head group to be out competing it in the cell, given, you know, because the power of a PROTAC is so efficient to reduce the amount of protein, it's not really going to be a problem in the cell. But it's more of a problem if you're looking at just a simple direct binding assay like spectral shift.
I might just add on to that is that I'll come back to the reaction optimization that we run before doing the D2b synthesis itself. So, you know, we'll always try and optimize the reactions for that kind of global set before we go in with the D2b synthesis. And if we can't achieve good conversion, acceptable conversion, then we'll come back and rethink it before we sort of charge up the garden path with it.
Exactly. So it's a combination of things.
Yep.
It's all about going into a well-validated assay with high quality plates of compounds.
Thank you very much. And what's the maximum plate size for D2b technology at DomainX?
So we use 384 well plates pretty much exclusively. There is new literature coming out, or newer literature coming out, describing 5036 well plates, and there's pros and cons to both. In principle, you can get through a higher throughput with 1536 well plates, but you can often encounter other bottlenecks, principally from a chemistry side. That would be UPS-CMS time. So we use an optimized LCMS run where we've sort of kind of really pushed that that gradient phase to just a 2-minute run. But when you're looking at kind of 1536 well plates, you're still looking about 3,000 minutes worth of UPLC time, and in that example, you may well have been better doing 4 384s sequentially through the week to avoid that kind of new bottleneck that's been introduced. But there's pros and cons to both.
Absolutely. And also lots of assays are not miniaturizable down to 1536. So all you're actually adding in that way is more issues around liquid handling, whereas, you know, pretty much everything we do is at least 384 well compatible.
And on that point, so we also touched upon, so you displayed the workflow, the typical workflow, and beginning with the assay compatibility. Are there any that are not compatible with D2b?
So far, we haven't identified any. Again, we always do it on a case-by-case basis because some proteins and cells are going to respond differently to others. So that's why you have to do it every single time, even if you feel like you're repeating yourself. Now, you absolutely have to do it every time to make sure that you're not going to be, you know, getting false positives, false negatives, and wasting a lot of time and resources.
Excellent, thank you. Great range of questions so far. Thank you so much to the audience for contributing. There are still a few minutes left, also in case you want to use this chance to talk directly with our experts online today. I should say, if anybody would rather speak and ask their question rather than type it, then more than happy for you to do so. There is a reactions button, and I can also look down the list of participants if you want to raise your hand to do that. So apologies, I didn't make that clear at the start. So I'll just take a quick scan in case anybody do feel to raise your hand. Oh, another question's come through. 1 second. Do you use spectral shift to triage compounds before going into a hit degradation screen?
You did in this case, but it's not it's not obligatory. One of the nice things about spectral shift is that you can do it so quickly and relatively cheaply compared with buying a high bit line, that it's very, very attractive. And it's probably even better for approaches like, elaborating fragments and, which is some of the things that we're working on at the moment. But yeah, it's not obligatory, but it is a nice thing to run.
Jonik, is there anything further you wanted to elaborate on in terms of the types of chemistries that you've built into the D2b workflow, or have you covered everything in that regard?
Yeah, sure. I mean, I can just quickly touch on the sort of chemistries that we validated in plate format. So amide coupling, of course, that's first and foremost in the literature. As I mentioned, we've been looking at palladium catalyzed transformations, and so we've successfully translated both Suzuki and what called CN cross couplings to plate-based format. We've also done reductive laminations, and we're particularly proud of a two-step amine to azide to azole kind of copper click chemistry, as well in that kind of DP format. And again, we've taken each of these into some of our common assays to validate those conditions are successful. And so far, we've had some great success.
Perfect. And I've just seen Natalie's popped a contact email in the chat box for anyone who'd like to find out more about the D2b Product Toolbox. So that's fantastic. Thanks, Natalie. And yeah, once again, just thanks to everyone for joining. Of course, thank you to the DomainX team for that stellar presentation and that really insightful kind of presentation you gave on how you're helping scientists to accelerate their research. So there will be a follow-up email to those online. So you should be able to catch up on the key learnings from the webinar, and do share with colleagues as well if you think they have missed something they would find useful and relevant. Also to add, just to let you know, a date for your diary. So there is another DomainX webinar on the 22nd of October. That's a Tuesday again, and that's called Tackling the Challenges of Membrane Protein Purification, Enabling Drug Discovery Programs. So that is a separate registration. So do ensure that if you'd like to join that one, that you register for that separately. It can be found on the One Nucleus website. Any questions, then don't hesitate to get in touch. So with that, I think I'll bring the session to a close. Oh, there's just one further comment. Any experience with simple automated synthesizer for D2b? Sorry if I've not pronounced that correctly.
No, that's absolutely correct. Yeah, the simple systems are a really nice system. It's something we've been keeping an eye on, and we may look to invest in the future. As of right now, we've tended to go with our own plate designs and kind of own way of adding in those conditions. But no, it's a really cool technology. I can well see its potential application to D2b.
Fab. Okay, brilliant stuff. Thanks, guys. That was really interesting, and thanks everyone for joining our session. And we hope to see you online for the next one. Have a great afternoon, everyone.
Thank you.
Take care now. Bye.